today's journal entry is where we're going to transition from talking about a genetic disease to talking about a chromosome disease. Now we're going to do our journal entry just a little bit differently so that we can work a little bit more independently. What I'd like for you to do is copy what is in black and answer the questions, filling them out. It turns out that you should actually know a lot of this. So if you need to go back, meiosis is going to be in section 2.3. So we're going to see that a lot of this information is from 2.3. So if you will pause this and go back and look at exactly how maybe you could find the words that go in here. And then when you're ready, just press play and I'll kind of go over the journal entry. So if we're ready to talk about the journal entry, what we want to see is a real example of someone who was born with a chromosomal disorder. So we were studying gene diseases or genetic diseases for a long time. Examples are hemophilia, red-green color blindness, cystic fibrosis, Tay-Sachs, Huntington's disease, sickle cell anemia, and PKU. Those are our 7 genetic diseases that we studied. Some are dominant, some are recessive, some are codominant, some are sex-linked. But it turns out that you can also have a disease where there's something wrong with the whole chromosome. So a real-life example of this is Sarah Palin. She was once the governor of Alaska and ran for vice president in 2008. She did have a son whose name is Trig, and he was born with a disability. But his disability wasn't a gene, it was a whole chromosome. So what is a chromosome? If we stop to think about how are chromosome disorders different from gene disorders? A chromosome is a section of coiled DNA. So remember, you have a lot of DNA and it's going to be coiled up into chromosomes. And humans normally have a total of 46 chromosomes in your body cells. So every body cell should have 46 chromosomes. What can sometimes happen is sometimes you can end up with the wrong number of chromosomes. Normally, during meiosis, the chromosomes will double and then divide, going from a diploid cell to 4 haploid cells. Each gamete, or haploid cell, will then have 23 chromosomes. The problem with this is this is not Trig's karyotype. This is not actually a picture of his chromosomes, but just of someone who has Down syndrome. This disorder is Down syndrome, and it's called trisomy 21. And so we're going to go forward now to our notes. We're actually going to page 62 in our notes. Page 62. If you'll go to page 62, we're going to talk about 3 different types of chromosome disorders where you have too many or too few chromosomes. You might be able to answer how a gene disorder is different from a chromosome disorder now, or you might need to go back and answer it later. But if you'll go to page 62 in your notes, and this is on chromosomal disorders. So if you need to pause it so that everyone can get to page 62, feel free. What we're talking about on chromosomal disorders are these are disorders that are inherited due to problems with the entire chromosome. It's caused by a mistake in meiosis. If you'll circle meiosis and write production of sex cells. Now sex cells should be haploid. Sex cells should be haploid. However, which means we're pulling apart the chromosomes, which we can kind of see here in this picture. Sometimes there's a mistake, and that mistake is called non-disjunction. That means fail to separate. So we should be going from 46 to 23, separating the pairs so that each parent only gives half. So the sperm has half and the egg has half, and they come together and they're a whole again. But non-disjunction is when we don't separate, we don't go to exactly half. And if a sperm has 24, which is one too many, or 22, which is one too few, or same with the egg, too many or too few, then we get something called aneuploidy. And aneuploidy means an incorrect number. If you'll circle U, if you remember U means true, and if you'll circle and means not, and if you'll underline ploid, ploid means number of chromosomes. So aneuploidy literally means an incorrect number of chromosomes in a fertilized egg or in a zygote. So normally we should have 46. So the normal number in humans, 46, but we might have too many or too few. So we're going to look at some examples of that. And here we see the picture a little bit bigger. The first example that we're going to look at is called Down syndrome. And Down syndrome is caused by an extra chromosome number 21. We call this trisomy. So if you'll circle or highlight tri and write 3. Normally our chromosomes come in pairs. So if you look at this picture, which is kind of similar to the picture that you have, trisomy normally pairs, so 2, 4, 6, 8, 10, 12, 14, 16, 18, 20, all the way up, we should have 46 in a human. But it turns out that sometimes we have extra chromosomes. This picture you don't have, but it does show 3 number 21s. Down syndrome will cause mistakes through the whole body because it's not just a tiny gene, one section, it's many genes on a whole extra chromosome. So we see problems over the entire body of the organism. You usually are going to see certain characteristics because there's too many proteins and it reacts in a certain way. Just like if a body has too much sugar, everyone might get diabetes and have the same symptoms. Here we have too many genes, and so these people tend to have very similar symptoms. It's not quite understood why these are the symptoms because there's so many genes. We don't know the answer to all of that. But Down syndrome, I do need you to write, is autosomal. It doesn't have to do with the sex chromosomes. 21 is not the sex chromosomes. The sex chromosomes are the 23rd pair. So Klinefelter's and Turner syndrome are actually problems with the 23rd pair, or the sex chromosomes. Klinefelter syndrome is you are a male, having a Y makes you a guy. So they are male, but they have an extra X chromosome. Now this does not mean that they make sperm and egg. In fact, it means that they're sterile. They don't have both parts. They can't make both sperm and egg. They are phenotypically, they show up as a male, but they're not able to develop certain secondary sex characteristics. So they're not going to have facial hair. They are going to have the main parts, the main organs of a male, but they're not able to actually make functioning sperm. You can use testosterone therapy to treat, but they will not be able to ever have offspring. And so this is a problem where there's an extra X. So XXY is Klinefelter syndrome. The next is called Turner's syndrome. Turner is where you are XO. So you're not XX, you're not XY, you're just XO. There's nothing there. And this is a female, there's no Y, but they don't have enough information to actually make the eggs. So they are going to be female, but they are also sterile or infertile. They can't produce offspring. And all of this has to do with a mistake that happened in the sperm or the egg. So it's not the offspring's fault at all, and it's not that the mom or dad did anything wrong. There was just a mistake in making the sperm or making the egg. We are going to go ahead and take a little bit of notes about nature versus nurture. This is the idea that some things you inherit and some things are your heredity. So keeping in mind that you can get certain DNA, but that doesn't mean you'll get certain diseases. So you might have an increased risk of a disease, like an increased risk of cancer, but that doesn't mean you'll necessarily get it. You could also not have a cancer gene, but end up getting cancer because of things in your environment, such as smoking cigarettes or being exposed to the sunlight. Scientists debate which one has a greater impact. Are your genes more important, or is your environment more important? A really interesting way to study this is by studying identical twins, because identical twins have the same DNA. They can sometimes be raised apart, which will allow us to study how their environment affects them. We can look at the diet, and we can also look at environmental toxins. If you need to pause it to get caught up, go ahead and press pause. Otherwise, we're actually going to go ahead to page 68 in our notes. So if you need to pause it, you can do that. What we're looking at here is actually the examples of how we are going to detect genetic disorders. And again, you might need to pause on each slide if we're having a little trouble going as fast as I speak, trying to fit it all into one video for you. If we want to detect genetic diseases, we can actually go see a genetic counselor, and that's something that you are going to be today. Today you're going to offer advice, acting as a genetic counselor, helping parents determine the chance of passing on a genetic trait, or helping to determine if the baby is going to be normal. And so you can use a sonogram or an ultrasound to get a picture, and most people are used to seeing these pictures. They can evaluate the baby's growth and development, so they can see if there are any physical abnormalities. They can also do blood tests to look for certain proteins. For example, if we want to know if a baby is at risk for Down syndrome, Turner, spina bifida, you can actually do a blood test. The other thing you can do is an amniocentesis. And an amniocentesis is where you remove amniotic fluid. Amniotic fluid is the fluid surrounding the fetus. Some of the fetus's cells will have come off, just like your skin cells naturally come off. And you can use a needle to pull out some of that fluid, and then we can actually look at those cells. And so a karyotype is a picture of the chromosomes. And from that, we can identify Downs, Klinefelters, and Turners because they are all chromosomal disorders. So if I look at a karyotype, I can see a chromosomal disorder. I can't see a gene disorder, so I can't tell if you have red-green color blindness. So what you're going to do today is look at some karyotypes and identify if a baby has Downs, Klinefelters, or Turners. You might want to use page 68 to help you. You can also do chorionic villi sampling. You can do this when the fetus is a lot younger. It's where you're going to take some of the surrounding tissue from around the baby. It is a little bit more dangerous if you do that. If we want to try to prevent or treat a genetic disease, we cannot cure them. Right now, we really don't have any ways to fix your DNA or fix your chromosomes. We don't have tiny little scissors or scalpels that can get into cells and change the DNA. We are going to talk on Monday about how to use genetic engineering to possibly replace defective genes. But we can give pain medications to help relieve symptoms. We can give occupational therapy, which is like physical therapy. We can give blood transfusions, and we're working on trying to give gene therapy. So where you're going to go from here is you're going to work on an assignment called Genetic Counselor. And there is a little bit of reading that you want to do to get some background. And then at the very end of class, you'll do an assignment with pedigrees and with the karyotypes. And you'll today be a genetic counselor.